Good morning, everyone. I'm going to talk a little bit about glyphosate-resistant kochia this morning, and I'm going to start off talking a little bit about resistance in general, and then move on to the kochia story. So, first of all, though, I want to acknowledge the other people that are involved in some of this work. Hugh Becky with Agriculture Canada in Saskatoon, Linda Hall with the University of Alberta, and she is a graduate student, Ryan Lowe, working on this project as well. So just want to get us all on the same page in terms of what we mean by resistance. It's the inherent ability of a plant to survive following exposure to a rate of a herbicide normally lethal to the plant. So we use this herbicide, it would kill all of our weeds, and then something changes, so those weeds are now surviving. Cross resistance, it means that we have only one resistance mechanism in that plant, but it confers resistance to more than one herbicide. So, and usually it's a family of herbicides. So in my example here, if we have group 2 resistant weed or group 2 resistant kochia, then that means it's resistant to a number of herbicides in that group 2 family of herbicides. And so that means Express, Odyssey, Solo, Everest, so it can be many of them. What we mean by multiple resistance is when a weed species then has 2 or more resistance mechanisms. And so in our case here in southern Alberta, we've had group 2 resistant kochia for many, many years, and now we have the new group 9 or glyphosate resistant kochia appearing in southern Alberta. So that kochia now has 2 mechanisms. What we think in terms of how resistance develops, there is that one plant in a population that is resistant, so it's present at a very low level. And what we've learned over the years, that might be a ratio of 1 in a million or even 1 in a billion plants is naturally resistant to that herbicide. We can't see it in those numbers. We don't know it exists. We spray the herbicide, and all the other plants are killed, and that red plant there, that red triangle, he survives, sets seed, obviously. That happens the next year, happens the next time we apply, the next time. And then over a number of years, and that can be short, that can be 5 years, or that might be 30 years, we just get a shift in the ratio of the population where that rare individual becomes the dominant individual in the population. And then we then have resistance that you can see in your fields and is there at a practical level. Now, some herbicides have a greater tendency to develop resistance than others, and a number of weed researchers in Western Canada sort of came up with this resistance triangle. So at the top here, okay, that's going to show up, I think. Showing up on the other screen here, yeah. So at the very top, this would be group 1 herbicides and group 2 herbicides. They have the highest tendency to develop resistance, and we say they would be less than 10 applications, we might see resistance developing. So group 1, if you probably all are aware, but it's many of our wild oat herbicides, such as Horizon and Achieve and Post, etc., and group 2 I mentioned before. So certainly we saw group 2 resistant kochia in 5 applications of a Glean or Ally treatment way back in the 80s. So it happened very quick. Wild oats, I know of wild oat populations that originally were selected for with Hograss and Puma that occurred after 7 or 8 applications of a herbicide. So that can happen very quick. Other ones, group 5 here is our atrazine, and then we go down to moderate, that would be Treflan, Avedex, and then the very lowest level here, group 4, is our phenoxy herbicides, our 2,4-D dicamba, group 6 is bromoxynil, group 9 is our glyphosate, and then group 10 is Liberty over here. So we think about glyphosate. It has a very low tendency to develop resistance. You know, we've been using glyphosate for a lot of years. We would probably suggest it requires more than 20 applications, but you know, we're sort of new in the glyphosate story, so don't really know that. I guess where glyphosate resistance has been around a little bit longer in Australia, I don't think they have any examples where they've used glyphosate less than 30 years. So it is a low tendency to develop resistance, but you know, if you use something enough, eventually you may get there with some weed species, maybe not other weed species. So that's the herbicide story in terms of some are more likely to get resistance than others. Now, the weeds also obviously play a big role in here, and so some factors come in terms of where you're most likely to get resistance or when it may happen. So the first one is just simply the distribution and the density of the weed. If the weed is very common in our area, widely distributed at a reasonably high density, then you have a greater chance of getting resistance. And resistance is very much a numbers game because it goes back to that 1 in a million or that 1 in a billion plant that may be naturally resistant before we apply the herbicide. So the more weeds you have out there, the more likely you're going to have that possibility. And then some weeds are more genetically diverse than others as well, so that means that they have a greater chance of having that resistant gene. And Kochia is actually a pretty genetically diverse plant. It's also an outcrossing plant, so that means it has gene flow from one plant to another and can have this spread of genes a little bit more quickly in that situation as compared to a self-pollinating weed species. And then high seed production. So if we have that resistant plant, and as I showed you in the earlier picture, if he's the only one that survives and all the other ones die, if that plant only produces 10 seeds per plant, it's going to take a lot of years before it becomes the dominant one. But if it produces a lot of seed, that'll happen more quickly. So what do we know about Kochia? It's been around here for a long time. I'm sure you all are very familiar with Kochia. You may or may not know it's a C4 species, so that's like corn. It's tolerant of heat, drought, and obviously tolerant of salinity too, so that's a big factor here in southern Alberta. One of the unique things about it, many of our C4 species, we would think that they require higher soil temperatures for germination. So for corn, we would say, okay, it really wants 10 degrees Celsius to germinate, but Kochia is quite the opposite. It germinates at very low temperatures, and it's often one of the first weeds we'll see in the spring, right, that's early emerging. But it will continue to emerge throughout the rest of the growing season as well. It germinates small seed, germinates at shallow depths, and produces lots of seed, and has a tumbleweed seed dispersal mechanism. So that's, stem breaks off ground level and blows across the land. So, and that's its inherent mechanism of dispersal. Low seed dormancy, and which means low seed bank persistence in the soil. So that can be a good thing if you have the problem and control the weed. Perhaps you have a chance of growing out the weed seed bank and depleting your problem over time. So, and it, we think of Kochia being a weed, I guess, of southern Alberta and southern Saskatchewan, but in recent years it appears to be sort of moving north, and perhaps with our warming climate. So Kochia has developed resistance to other herbicides in the past. As early as the 1970s, there was Kochia that was resistant to the triazine herbicides. And they're not widely used in our cropping systems here, but they were in other parts of the world. Group 2 resistance, as I said earlier, was identified in the 1980s. And we have so many group 2 herbicides. So if you go look in the blue book, the Guide to Crop Protection, you'll find that's 17 herbicides. So it's not, we say group 2, but that's 17 herbicides that will not kill Kochia in southern Alberta. And the reason why I say that is 90% of Kochia in southern Alberta is group 2 resistant. So you just need to sort of know that unless you can prove otherwise, none of these compounds like Express or Odyssey or Everest or whatever, none of these are going to work on your Kochia in southern Alberta. So think about something else. And then group 4, this is pretty rare. We don't have any resistance to Banville or 2,4-D in our region, but Banville resistance has been confirmed in Montana, Nebraska, and North Dakota. So that's my other message today. Many of you are going to use dicamba or Banville to control your glyphosate-resistant kochia, and so that's good, but you shouldn't do that over and over and over again, or 5 to 10 years from now we'll be talking about Banville-resistant kochia in southern Alberta. So now we're going to start talking about the glyphosate story. So glyphosate-resistant weeds, they're relatively new to us, but they're not totally new around the world. There are now 24 weed species around the world that are resistant to glyphosate, and they occur in North America, South Africa, Europe, Australia, China, so it is out there. Most of this has happened in the last 10 years, so it's relatively recent in that regard. And you'll see that there's almost an equal breakup of whether they're grass weed species or broadleaf weed species. In Canada, it's relatively new. In Ontario, it was found 2 or 3 years ago. They have 3 broadleaf species. They have giant ragweed, common ragweed, and horseweed. And then we have our kochia story, which is the first case in Western Canada. So now we're going to get into the kochia story here. This is a kochia distribution map in North America, and the darker the color in the center here means that it's the highest densities of kochia. So you can see that kochia is very much a weed of the Great Plains of North America, the drier regions of the U.S. and merging up into Canada here. And so where are we going to get resistance to a weed? I said earlier, we're going to get resistance where that weed occurs the most. So it's no big surprise that these stars indicate where kochia resistance was first found. It's right in that dark red, that dark orange region of where kochia exists. You're not likely going to get kochia resistance here in California or somewhere else because there's not a lot of that weed out there. So anyway, first confirmed in Kansas in 2007, and then several other states in subsequent years. So this is a photo from Kansas, taken by Dr. Phil Stallman from Kansas State University that did some of the early work on kochia resistance in Kansas. And this is one of the things that they were noting in their fields, is that they often saw this zigzag trail of kochia plants that had survived glyphosate applications. And of course, this is just the tumbleweed nature of this weed. Stem breaks off, blows across the field. It's dropping its seed as it does that, and then you end up with sort of this linear but sort of zigzag trail the following year. And so, I've known Phil for 25 years, and I had saw his presentation a couple of times and talked to him about what he was doing there. So we were aware of that, and that's a good thing that we often collaborate with people in other countries and on research problems. So then, this is us. So I got a call in August of 2011 from a grower, and said he had some problems in a chem fallow field, and we start off with the usual conversation of what rate and what water volume and blah blah blah, and then he sort of got into it. He says, well, it's only one weed, it's kochia. And so then I guess I became a little more alert at that point, and then he started describing his field, and I said, I just sort of thought to myself, okay, this doesn't sound very good. So I phoned up Hugh Becky from Saskatoon, and we went out together and visited his farm and a couple of neighboring farms. And so, what we saw is these linear trails across the field of kochia plants, and pretty much everything else was dead. And he sprayed 1.25 liters of Roundup here a couple of times, and these plants were surviving. Another field, this is what it looks like again. Sort of, you tend to have these trails of surviving kochia plants. But not always. Sometimes you just see you have a healthy plant a couple plants here, and then, you know, and then all around that are dead plants. And you think this one should be dead as well, but it's not. So those are, you know, the symptoms we saw, and I guess it's a thing that all of you should be, you know, sort of aware and perhaps looking for on your own farms, on your neighboring farms. So what we did is we, obviously the farmer wanted to control those plants, didn't want to let them go to seed. So we collected plants and put them into pots and brought them back into the greenhouse for testing. So, so this is in the bottom here, this is Hugh Becky from Saskatoon, myself, and up here in the black t-shirt is Ryan Lowe from the graduate student from University of Alberta. So we collected plants from those 3 fields and put them in a bunch of pots here and took them back to the greenhouse. And we let them mature and collect mature seed on those plants, and then we planted that seed and tested that and let those plants grow, and then we sprayed them to test to see whether they were resistant. And this is just our growth cabinet sprayer. So this is what we ended up with in our greenhouse studies. So we had 3 Alberta populations from those original 3 farms that we collected plants from. We were very fortunate enough that we were able to go back to Phil Stallman and ask him if we could get some populations from Kansas that we could work with, because we were very interested in comparing our populations to the U.S. populations, because they had already, you know, they had been doing the research 3 or 4 years ahead of us, had characterized their populations, knew something about it. So you know, it made sense to sort of do a comparison. And then we had 2 susceptible populations, one from Saskatchewan and Kansas. And then we just sprayed 9 different rates of glyphosate, you know, from a 0 up to 2250 grams active per hectare. So if we say 450 or our half liter is a 1x rate, we went from sort of a 1x to a 5x rate of Roundup or glyphosate. Now I'm going to show you a few pictures. I realize this picture is not very clear. I've got better pictures here. But what we've got here, we can pretend, I think whoever set this up probably has some Chinese background, so we're going to read from the right to the left. We're going to go backwards. So this is our 0 rate of herbicide, and we're going to our high rate of herbicide on the left-hand side. So this is our susceptible population, and you see we get up into this range is our half liter rate. We've got a pretty good kill on our Kochia. This is one of our Alberta fields. You see that the plants are surviving pretty well, and even at the highest rate, they look pretty sick, but they're surviving. And the trend was very much the same as the Kansas population in terms of what rate was required to get what type of control. And these are the better pictures that I was referring to. So this is our half liter rate of glyphosate, 450 grams active per hectare rate. And you can see that the 3 Alberta fields are labeled F1, F2, F3, and the Kansas population, you see they're surviving pretty good at that. This might be what we may call a 2x rate, and they're stunted but still alive. And then we get up to the 5x rate, and they look pretty sick, but they're not completely dead at that rate. So then we throw this data on a chart, on a graph here. And what we're looking at is survival on the y-axis, and then the x-axis is just increasing rates of glyphosate. And the 2 green lines are our susceptible populations, one from Hayes, Kansas, and one from Hanley, Saskatchewan. You see that, you know, relatively low rates of glyphosate, we're getting a good kill on those plants. And we go look at the resistant populations being up here, the red lines being Alberta, and the blue ones being Kansas. And you can see that there's a lot of overlap. Are they responding very similarly in a very similar manner, and they're requiring higher rates of glyphosate. So what we do to calculate this resistance factor over here is we just take the amount of herbicide that would be required in the resistant one and just divide that by the amount of herbicide that is required for the susceptible population, and then we calculate this value. So that's what that resistance factor means. So it's just a ratio of how much herbicide, more herbicide is required to get an equivalent rate of kill or an equivalent rate of control. And so for the Alberta populations, we needed from 5 to 7 times more glyphosate to get an equivalent rate of control. And for the Kansas ones, it ranged from 4 to 6. So we're sort of in the same ballpark. So all this stuff here was done in the greenhouse, and so we're pretty confident of those results, but we wanted to go out and do it in the field as well, just to see if is it really the same level or the same situation in the field. So I did a field confirmation study at Lethbridge in the summer of 2012. The same thing, we had the same seed from those 3 Alberta populations, planted them in the field, and we applied the same 9 rates of glyphosate, did the whole same thing in the field, and we came out with a resistance factor of 6.2. So very much in the same ballpark. So we're very, very confident that resistance is real, and that this is sort of the level of resistance we're talking about. So in that 4 to 5 to 6 type of range of resistance level. So that's not really high resistance. I mean, that's real for you as a farmer and for all of us in the ag industry. That's a significant level, but it is sort of, in terms of resistance, is a relatively low to moderate level of resistance. If we were to talk about group 1 resistant wild oat, we often talk in the range of 20 to 30 to 50 times. And group 2 resistant kochia might be 50 to 80 times resistant. So very high levels of resistance. So this is a much lower level, but it's still biologically real and is significant on your, for anybody in the field. So that was the original 3 fields. Now where do we stand in terms of how widespread is this? We don't really know that story, but I'll tell you what we do know. So we collected those original 3 fields in August. We went back in the fall of 2011, and we collected, we went to 46 sites within a 20-kilometer radius of those original 3 fields, and we found that 7 more fields had glyphosate-resistant kochia. And then in the spring of 2012, there was another population that was delivered to me in the Turin area, and that was also resistant. So, it's already spread a little bit, or spread isn't the right word because we don't know where it originated from. So, and we're certainly not blaming those original 3 farmers or those farms. There's a very good chance it did not originate on those farms. It could have came from another farm, it could have came from an oil and gas site, it could have came from a ditch, it could have came from a railroad. This one farmer was just actually good enough to give me a call, and I thank him for doing that because we need to be aware of what's going on. There's no use ignoring the problem. So in the summer of 2012, I had several more populations delivered to me for testing, and some of them were right from the Medicine Hat region here. So they sort of had the telltale signs of plants were living and other plants beside them were dead, and some of them had sort of trails across their chem fallow field. So the odds are pretty good, but we don't know yet. They're being tested. We had to, same thing, we had to let mature seed and plant it. They're being tested right now, so we'll have results later on this winter. Hugh collected some from Saskatchewan. I say 4 here, but actually I think we talked on the phone just a couple days ago, and he said, well, that number is more like 8 or 10 farms that he collected seed from. So we're evaluating all of those. And then we did a totally random survey in southern Alberta just this last fall. So just south of the Trans-Canada Highway, we went to 300 sites and collected seed, and just to sort of get a better idea of the distribution of that. So it would be nice to say that it's a localized small problem, and we don't really know otherwise, but it's probably a little bit more out there than what we think. So now the good news story here at the very bottom is, well, good and bad news, I guess. All of the ones that have confirmed to be glyphosate resistance are also group 2 resistant. So that was no surprise to us because, and it should be no surprise to you because 90% of all kochia in southern Alberta is group 2 resistant. So, and we just confirmed that again in all of our trials. But the good news is here is that they were all susceptible to group 4 herbicides, or most importantly, susceptible to dicamba, which is a very good, news story for us because, as I told you, some of the U.S. states already have dicamba resistance. So, we're sort of hoping that we're not there already. So the resistance mechanism, these are early days on that. We don't really know. Again, we have good collaboration, as I mentioned, with Phil Stallman in Kansas. We also have good collaborations with Phil Westra at Colorado State University. We sent some of our seed down to their lab in Colorado. And one of the things they're finding here is that all of their populations and all of our populations that we sent down there have this potential mechanism of gene amplification. So it just means there's more genes that are coding for the enzyme that glyphosate inhibits. And it's sort of like a dilution effect. If you have more enzyme in the plant, maybe there's not enough glyphosate to knock out all of that enzyme, and the plant can survive. So that's probably has something to do with it, whether that's the sole mechanism or whether there's other mechanisms out there. So early days on that work. So now I'm going to show you, just coming towards the end of my presentation here, and that's good. We'll still have time for some questions. We're just starting, some of our research in this area. So this is early days. So I'm going to show you some data here, but I just have to caution you, this is only one year's data, and it's only one site. We tried to do this in Saskatchewan, but Hugh's experiment was flooded out this year, so he lost everything. So only 1 year and one site, and so very, very preliminary data. And it was almost meant to be just a bit of a screening trial in terms of what herbicides would we expect to be effective and what wouldn't be. So we did this in a non-crop situation, so I guess it was simulate a chem fallow or a pre-seed burnt ion type of situation. And that's important because some of these herbicides would work better. If you had crop competition that was helping them out, they would be more effective than if they were just in a non-crop situation. We sprayed the Kochia when it was 5 centimeters tall, 6 centimeters in diameter, so maybe a little bit larger than what you might do in some situations, but if you're in a chem fallow situation, probably many of your plants would be larger than what they were here. So, and our density was 30 plants per square meter. And again, so then this was a seed that we had collected before, so we know they're group 2 resistant, and we know they're group 9 resistant or glyphosate resistant. So the first, and then we have the control rating. So the first thing I want to talk about here is glyphosate, and we got 35% control. And the first thing you say, well, okay, that must be a mistake. You just told me these plants are resistant to glyphosate. Well, the first thing is we still get just a tiny little bit of suppression at that rate of glyphosate. So that comes up in my control ratings. And the other thing is the population of Kochia that we planted was not totally homozygous for that resistance trait. So it means that we had some of our seed, 5 or 10 or 15% of that seed was probably still susceptible to glyphosate. So that's why we come up with glyphosate actually giving us a little bit of control here. So then the other thing you need to note here is that then glyphosate was included in all treatments. So all of these next treatments are glyphosate plus cadet, glyphosate plus partner, glyphosate plus Buttrell-M. So there's glyphosate in there, and you know that from glyphosate itself is giving you 35% control. So if we were to look at partner, it's 60%. So really the partner was maybe 60 minus 35. So that gives you some idea of how good those products are. So I've broken the table down into 2 parts. So this is the first part of the table, and I would say that really none of those herbicides in there are very good. You're not going to be happy with any of them. Because they're 75% control or less. So just a couple of things maybe to point out. Cadet is an experimental herbicide, so we'll continue working with that, and the company suggests we need to go to higher rates. I think probably most of you know that really, partner Buttrell-M are not great on Kochia, never have been. What may or may not be a surprise to you is that 2,4-D is not very good on Kochia. And never has been either. This is not a resistance thing or anything that's sort of changed. I mean, we think of 2,4-D as being good on Kochia, but it's often been mixed with something else when we apply it in our fields, right? So it's often been mixed with dicamba or mecoprop or dichloropropyl or maybe something, something else. So you're not going to be happy using 2,4-D as the answer in your chem fallow fields or maybe even your pre-seed burn down. So you just need to know that. Banville, dicamba is good. This is what you might consider the low rate, the 140 gram rate. If you're an old guy like me, that's the 2 ounce rate, 2 ounces per acre rate. So that's sort of a borderline rate, and you probably want to go to a higher rate. In a chem fallow field, for sure, you're going to go to a higher rate. And I'll show you what those numbers are. So this is the second table of the same experiment, but then I would say that really all of these ones in here are probably pretty good for you and something you want to consider. So Fluoxapure is very good on Kochia, so that's all of your Attain, Prestige products, all the mixtures it's in. You know, that was giving us 85% or 90% control across the board. Infinity is a relatively new herbicide, and that worked very well. And then I'll show the Banville. You know, we get to higher rates. We're getting up to 90 and 98% control. So you just get up to a little higher rate of Banville. It is going to be a good treatment for you. Optica Trio, also very good. BASF's new compound, Distinct, is also a very good product, giving us excellent control. And then the Clean Start Authority here. You know, this would be the low rate of Clean Start, I guess. You know, there's sort of 2 rates for that, and Authority, it's registered at 105 to 140. So this is really just a half rate of Authority or half rate of sulfentrazone. So sulfentrazone is dynamite on Kochia, more of as a soil active sort of compound. It gives you a bit of residual control. So it's something that may fit into your plans if you can find a way to use that. And of course, sulfentrazone has a fit in some of our pulse crops. So the good news here is that we do have a number of herbicides that will control glyphosate-resistant Kochia, and they come from different chemical families, different herbicide families. And so that's going to be important to think about how we're going to manage this problem for the next 20 years. What are we going to do? And so I already told you, I just don't want you to use only dicamba or only Banville for the next 20 years. So try and use some other compounds. So group 2, write them off. Nothing's going to work here in southern Alberta. The group 4s, we got choices. Group 14 is many of these new herbicides that have just become available. Group 14 herbicides have been around for over 20 years. They're not new herbicides, they're not new mode of action, but they are new to us in Western Canada. And we haven't used them a lot, so I think there's some good opportunities to use them. Saflufenicil is heat. I had a footnote in the previous table, if you notice, we applied heat and we'd expect it to be good, but we had an application error, so we didn't get any data on it. So any of these are worth considering. And then here's group 19, which is part of Distinct, and then Infinity is a group 27. So just think about trying to use different modes of action, you know, as you manage this problem going forward. So what additional weeds might we see in the future? We had some predictions a few years ago, and Kochia was in the top 3 weeds that we guessed as a weed science community, and unfortunately we were correct in that guess. It was one of the first ones we found. So what are our top 3 guesses now? Are wild oat, green foxtail, and cleavers. So I guess my message is just beware, especially when you're looking at those weeds. You know, if you see something that you think is not right, if you think a chance of resistance, then, you know, try and let somebody know. Where are we going? Future research. I said this is early days. We're just getting started. Hugh, Linda, and I are busy writing research proposals right now to try and attract money to do some research in this area, and depending on what grants we're successful with, some of these are some of the things we're interested in doing. We certainly want to continue on with identifying the most cost-effective herbicides. We want to do a little bit more in terms of kochia biology. I'd like to know better when do we have mature seed on a kochia plant. I can't tell you. If you can tell me, I'd like to hear from you, but it's very difficult to tell when that plant is mature. We know it's sort of later in the fall, but if someone came to me and said, okay, I'm going to terminate that plant on the 1st of September, do I have mature seed or not? I don't know. Is that September 15th? Is that September 30th? I don't know. And you can see some other things we're interested in doing here. So we certainly plan on doing this research for the next 3 or 4 years, and we'll have more information for you at that time. So this is my last slide, and I think if you haven't been to this website, you should jot down this website and go there. What happens here is you're just asked a series of questions, and you respond to those questions, and it's a risk assessment tool for you to determine what level of risk you may be at on your farm, or even a particular portion of your farm, in terms of developing weed resistance. And so, you know, the first things will be just simply how many times have you used glyphosate, but there's a number of other questions in terms of your crop rotation, in terms of your tillage practice, and in terms of those other things as well. So it's definitely worth a look-see and go there and play around with that site a little bit. So I'm going to end it there, and I think we do have a little bit of time for questions, at least a couple minutes here. So thanks for your attention, everyone. So thanks a lot. Bye. Questions? Sure. Try the mic. See if they've got it set up.
Yeah, I think it works. It's a little low for me, but try to pull it out. I had a question about the wheat, or the chemical resistant kochia. Like, is there also resistance, like a physical resistance? Like, I'm thinking of kochia that has, you know, the hairy leaves kind of thing, and I'm thinking if there's more hairs on the leaf, or if the hairs are longer, does the chemical penetrate through the hairs to get to the leaf surface? And if that's the case, then maybe you should try to include higher water volumes and maybe some sort of surfactants too, to see if that makes a difference, right?
Yeah. So that's very true. We've known, you know, for a long time that kochia is a difficult one to get uptake and penetration into the plant, and you know, that's sort of the whole factor of control, but it's not going to be enough to make a difference in this case. If the level was only a resistance factor of 2, or, you know, very, very low, maybe, you know, you could just go to higher rate, or you could use surfactants, you could do something else. But this is high enough level of resistance, it's not going to be enough to overcome that.
You made the comment about the cross resistance of group 2 and with glyphosate in kochia. Any knowledge coming from Kansas about group 4 and glyphosate cross resistance?
No, I guess the answer is no. As far as I know, I guess I don't know, but yeah, I'll leave it at that. I don't know. It's relatively rare, and where it was happening is really it came from group 2 resistance, which, you know, so farmers couldn't control their kochia with their group 2 herbicides, so they switched to dicamba, and some of that they were switching back 10 or 15 years ago, and that's when the Banville resistance occurred, because they switched en masse to using dicamba. So it's not because of the glyphosate story. So the answer is we don't know, but you know, they're not really connected. It just means if everybody just switches to using Banville, you're probably you have a chance to getting dicamba resistance. But they're different mechanisms, different genetics, different mechanisms. So just because you have glyphosate resistance, you're not more or less likely to get Banville resistance.
Yeah, and I'm of the same mindset. It's more, I guess, if that one genetic seed is out there where the 2 existed, and then I guess just to be aware of what could be happening down the pipe as farmers start adjusting. The alternative, or another follow-up question, was with regards to authority. When you mentioned about its longer season control. Vegan, I guess when you have longer season control, there's that selection pressure that continues on. If you just want to comment about how that relates to resistance development in a weed. Right.
So we used to think the longevity of herbicide persistence in the soil was a big factor driving resistance, and that came from the early days of the trizine herbicides when we were thinking of atrazine and simazine that had a long persistence in the soil, and we thought that was what was driving resistance, and it contributes to it. But then along came the group 1 resistant wild oat herbicides that have no persistence in the soil. And we got just as high a level resistance, and we got it in less years than we got it with herbicides that were persistent. So we sort of backed off that whole story and said it's not as important as we thought. So it's a little bit important, because what it means is it's not just the weed that's up, but then the weed that germinates a month later, you can also get control, and it sort of drives that resistance a little bit, but it's not as important as we used to think it was. So, but just to sort of throw out that sulfentrazone, it is persistent. It can be persistent enough that it'll hurt next year's crop as well, so you need to be aware of that. It is used in chem fallow in parts of the U.S., and works okay, but they've also had cases in a dry year where it hangs around for the whole year and hurts next year's crop. So you need to be aware of that on the whole authorities story as well. Yes?
On that list of other glyphosate-resistant weeds in the U.S., you had one on there called horseweed.
Yeah.
Is that what we call Canada fleabane up here?
Yes, it is.
So that's starting to show up in some field margins and stuff quite a bit, I've noticed the last few years.
As a weed here.
Yeah.
Yeah. Yeah, so it's certainly one that's, it's in other parts of the world. It's the one in Ontario as well, and other parts of the world. And so, so if you have it in your area or on your farm, it's one you should be watching out for. Because that's how we sort of use, these guesses are predictive. It's a bit of knowing, where it's occurred in other parts of the world, in that species, or maybe in that family of weeds, and is a genetically diverse weed species that has a tendency over the years that we've seen it happen with other compounds and stuff like that. So if it's around, then it's definitely one to watch out for.
Okay, we'll have to wrap up there. We have to keep carrying on. Jamie's giving me the evil eye, so I'd like to thank Dr. Blackshaw for coming, and he'll probably be around for a while this afternoon. So if you have any more questions, line them up at lunchtime. Thanks, Dr. Blackshaw.
Yeah. Thank you.